Welcome to the course of Comprehensive Molecular Diagnostics and Advanced Gene Explanation Analysis. So this is the week 6th discussion. I am Uditya from IIT Madras, and today we will be looking on different proteomics tools. Overview, importance and methods in proteomics, high performance liquid chromatography, mass spectrometry, differential in-gel electrophoresis, and protein microarrays. So coming to proteomics, what is proteomics? Proteome essentially means properties and activities of all proteins an organism makes in its lifetime. And naturally, proteomics is the qualitative and quantitative comparison of proteomes under different conditions. So why proteomes is necessary? So these are some of the points that are there. That mRNA levels is not something that we can directly correlate with the protein expression levels. Post-translational modifications affect protein functions. One gene can produce multiple proteins, and proteins, not genes, execute biological functions. So that's what makes proteomics essential. Then coming to types of proteomics, there are several types of proteomics. Expression proteomics, in which we study changes in protein expression. We have structural proteomics, in which we study the 3D protein structures, and then we have functional proteomics, in which protein interactions and functions are studied. The basic proteomics workflow usually has these three major steps, in which first step is sample preparation, in which protein extraction is done using lysis buffers. The common components of this lysis buffers usually are tris, HCl, NaCl, some kind of detergent, SDS or Triton X100, and reducing agents. After this, then the protein is separated. Then we have several gel-based techniques, gel-based approaches, and chromatography-based approaches. In detail, you can read this in this PPTs. And then after this are separated, we can go for protein identification and characterization. So this protein identification characterization usually are done by some common techniques like mass spectrometry. Then you have differential in-gel electrophoresis and protein microarrays. High performance liquid chromatography is one technique that we saw of protein separation under the chromatography based approach, in which it works on the principle of extension of conventional liquid chromatography using high pressure. So this uses smaller packing particles in metallic columns. And some of the components are solvent reservoir, pump, injector, column, detector, and recorder. Coming to types of HPLC, there are two types. One is called as normal phase, in which polar stationary phase and non-polar mobile phase is used. Another one is reverse phase, in which non-polar stationary phase. Example, C18 carbon 18 chain, and polar mobile phase is used. Coming to the applications in proteomics, these are useful in protein and peptide separation. When coupled with mass spectrometry, protein identification and quantification can also be performed. And purification of proteins, high sensitivity and throughput analysis HPLC can offer. Coming to mass spectrometry in proteomics, it works on the principle of mass to charge ratio of gas phase ions. Major steps that are involved is sample inlet. Sample is introduced in the inlet. It goes from ionization, acceleration, deflection, and detection. Some of the key ionization techniques are electrospray ionization and MALDI, matrix assisted laser desorption ionization. The mass analyzers are there. Two types have been mentioned here. One is time of flight, in which the flight time is measured, which is proportional to m by z ratio, mass to charge ratio. And then there is quadruple, in which electric fields, which uses electric fields to filter ions by mass to charge ratio. Applications are, it is used for protein identification and sequencing, structural analysis, and when coupled with LCMS or MS, it can enhance sensitivity and enhance selectivity and sensitivity. Then we have another gel technique, which is differential in-gel electrophoresis. In this enhanced 2D gel electrophoresis, it is an enhanced 2D gel electrophoresis technique. It uses multiple protein samples labeled with different fluorescent dyes, and the samples are run on the same gel, eliminating gel to gel variation. Workflow is, it uses sample labeled with cyanide dyes. Then the samples are mixed and dissolved due to electrophoresis. The fluorescence imaging at different wavelengths are done, and the image analysis and spot identification is something that is done. Mass spectrometry of differential spots then is performed. Advantages is that it has reduced gel to gel variation, higher sensitivity and quantitative accuracy, and simultaneous analysis of multiple samples can be done. Then another technique, the last technique of protein, which comes under protein detection, is protein microarrays. This protein microarray is a miniaturized assay platform with arrayed proteins on a solid substrate, and which enables high throughput study of protein-protein interactions. Types are analytical microarrays, reverse phase protein microarrays, and functional protein microarrays. So these three types of protein microarrays are possible, and each has its own utilities. These protein microarrays can have several other detection methods which can be used to analyze the assay. As I told, it is a miniaturized assay platform, so it can be label dependent, in which fluorescent dyes, enzymatic and radioisotopes are used, or it can be label free in terms of SPR, CELDI-TOF, MS. Coming to applications, this protein microarrays can have applications in disease diagnostics, biomarker discovery, protein functional analysis, antibody characterization, and drug development. Okay. That was a quick summary. ELISA and protein microarray. So ELISA usually are done in ELISA plates here. So it's kind of similar. The detection techniques are usually similar only. Like the assay platform is essentially similar, but ELISA is a kind of subsidiary as ELISA is enzyme linked immunosorbent assay. Yeah, protein microarray can be.
But like in microarray also, if we use fluorescent labeling, then we
Yeah.
need enzyme.
No, fluorescent it is directly. Yeah. You don't need an enzyme in terms of fluorescence.
Okay.
Okay. So yeah, we'll go to the problems. So you can put the options in chat, and we'll then discuss it. So we'll wait, I think, for 3 minutes each question, and then we can go ahead. As it's for the online also, on the people who will see videos too, so we have to do that. So what is the definition of protein? The four options are there. So the answer is properties and activities of all proteins that an organism makes in its lifetime. The proteome is defined as a protein complement to a genome, yeah, representing the entire set of proteins expressed by an organism at a particular time under given conditions. So the answer is properties and activities of all proteins that an organism makes in its lifetime. Which of the following agents is not commonly used in lysis buffer for sample preparation in proteomics? So the answer here is acetone. Yeah. So while trisacyl, STS and beta mercaptoethanol are commonly used components in lysis buffer for protein extraction, acetone is typically used as a precipitation agent rather than a component of lysis buffer. Common lysis buffer components include trisacyl, NaCl, triton X100, sodium dioxycholate, STS, EDTA, and reducing agents like beta mercaptoethanol or DDT. So the answer is acetone. Which technique separates proteins based on both isoelectric point and molecular weight? The answer here is two-dimensional gel electrophoresis. The technique first separates proteins. Yeah. The technique first separates proteins based on their isoelectric point. Yeah. Through isoelectric focusing in the first dimension, and then by molecular weight using SDS page in the second dimension. So you have two-dimensional gel electrophoresis providing a powerful resolution of complex protein mixtures. Yeah, so that's two-dimensional gel electrophoresis. How does ion exchange chromatography separate proteins? The answer is based on their charges. So ion exchange chromatography separates proteins based on the differences in their net charge based on the differences in their net charge at a specific pH. The stationary phase in the column has charged groups either positive or negative, which interact with proteins of opposite charge. So the proteins with the same charge as the stationary phase or neutral proteins pass through unrated, while oppositely charged proteins bind to the column and are eluted by altering buffer conditions, that is pH or salt concentration. Yeah. So it's purely based on the electrostatic interactions. What distinguishes high performance liquid chromatography from conventional liquid chromatography? So the answer is HPLC uses smaller packing particles and high pressure. So HPLC is distinguished from conventional liquid chromatography by its use of smaller packing particles in the column, which increases the surface area for interactions, and higher pressure to push the mobile phase through the tightly packed column.
बहुत मिर्ची है ना उसमें।
And good question, right?
This results in better separation efficiency and faster analysis time. So here it uses higher pressures up to 400 atmospheres, yeah, to force the mobile face through tightly packed metallic columns. Yeah, that's the difference. Cool. Which component of HPLC generates the flow of element from solvent reservoir into the system? The answer is pump. The pump is positioned at the uppermost stream of the liquid chromatography system and generates the flow of element from the solvent reservoir into the system. It must provide consistent pressure and controllable reproducible flow rates. Yeah. So this is essential for efficient separation of analytes. What is retention time in the context of HPLC? So the answer is time taken for a compound to travel through the column to the detector. Specifically, it is measured from the time at which the sample is injected to the point at which the display shows a maximum peak height for that particular compound. Yeah. So that's the definition for retention time. The time at which sample is injected to the up to the point at which display shows a maximum height, peak height at the for that particular compound. Okay. Which ionization technique is considered a soft ionization technique that causes minimal fragmentation? So the answer is electrospray ionization. ESI is a soft ionization technique that causes minimal fragmentation, making it suitable for analyzing fragile or large molecules such as proteins and peptides. In ESI, a liquid sample is nebulized in fine charged droplets that eventually form gas phase ions. In matrix assisted laser desorption ionization MALDI, what is the role of the matrix? So the answer is to absorb laser energy and transfer it to sample molecules for desorption and ionization. In MALDI, the sample is mixed with a crystalline matrix compound forming a matrix sample co-crystal. When irradiated with the laser, the matrix absorbs the energy and transfers it to the sample molecules, causing their desorption and ionization. What is the principle of time of flight mass analyzers? The answer is C. Ions are accelerated into a flight tube and their time of flight is proportional to the m by z ratio. So this mass to TOF provides high mass resolution which allows for accurate determination of m by z values, which is crucial for precise identification of ions. Yeah. So yeah, that's how they it allows for accurate determination of mass to charge ratios. Yeah. What is the purpose of the internal standard in DIGE? So the answer is to match and normalize protein patterns across different gels. Yeah. So the internal standard, which contains all proteins from all samples, serves as a common reference point across different gels, negating the problem of intergel variation and allowing accurate quantitation of differences between samples. Yeah. What is the primary principle of protein microarray technology? The answer is B. Immobilization of proteins onto a solid substrate to study protein interactions here. So protein microarray technology involves arranging proteins on a solid support surface such as glass slide or nitrocellulose membrane to allow miniaturized experiments for studying protein interactions. Which type of protein microarray uses antibodies as capture molecules to bind specific proteins from complex samples? So the answer is analytical microarrays. So also known as capture arrays, analytical microarrays use library of antibodies arrayed on a support surface as capture molecules, with each antibody binding specifically to a particular protein in a complex sample such as cell lysate. So the answer is protein microarrays. What detection method in protein microarrays combines solid phase chromatography and time of flight mass spectrometry? The answer is CELD, surface enhanced laser desorption ionization. Yeah, CELD can be seen as a combination of solid phase chromatography and time of flight mass spectrometry, where proteins of interest in a sample become bound to a surface before MS analysis. Yeah, so here you can see the process. So how it is bound, and due to the laser beam, then the sample ions are going to the mass spectrometry. Yeah. Which of the following is a label-free technique, label-free detection method for protein microarrays? So SPRS, yeah, surface plasmon resonance spectroscopy, is a label-free detection method that does not require the use of labels like fluorescent dyes or radioactive isotopes, which might affect protein activity. Other label-free methods include optical ellipsometry and reflectrometric inference spectroscopy. Yeah. In a DIGE experiment, the fluorescence intensity of a protein spot from a control sample labeled with Psi3 is 25,000 units. The same spot the same protein spot from a treated sample labeled with Psi5 has an intensity of 42,500 units. The internal standard labeled with Psi2 shows intensities of 30,000 and 32,000 units in the two gels respectively. Calculate the normalized fold change in protein expressions between the treated and control samples. first step is to normalize. You have to first normalize each to their each standard. Yeah, so normalized control equal to control divided by internal standard. Yeah, so this is 25,000 divided by internal standard is 30,000, so it comes out to be 0.833. Similarly, normalized treated equal to treated internal standard, it will be 42,500 divided by 32,000. Yeah, so finally the fold change here equal to normalized by normalized control. Yeah, the answer is 1.59. Okay, in a protein microarray experiment, the mean fluorescence intensity for a positive control spot is 12,500 units with a standard deviation of 750 units. The mean background signal is 500 units with a standard deviation of 150 units. Calculate the signal to noise ratio for this assay. So we have to calculate it using the formula. Yeah, SNR equal to mean signal minus mean background SD signal square plus SD background square. So 12,500 minus 500. Yeah. So that's all I had for today. If you have any doubts, you can tell.